whole thing?
when we're done.
Okay.
Yeah.
Under-
These anemones, shrimp.
Yeah.
We might need to slurp. Like, let's get some 6 batches one of those, and then we might need to slurp.
Okay.
Next.
Okay. What, do the brittle stars first?
Let's get some good shots first. Then
Yeah.
I think then
Yep.
batteries. Then mussels,
Okay.
because a lot of stuff will come with the mussels.
Okay. Yep. All right. So first brittles or shots then.
Do you want to shut out the panel?
What's that?
You want to shut out the panel and they zoom in and
I can?
Yeah.
Oh boy. That's a lot of responsibility. All right.
You see, you know what you want. So to the left, maybe rotate the head now to zoom out, right? Then to the right and zoom in.
Remember that we went through last week?
Yeah. Where did you see the brittle stars? Oh, they're down. Can't go any further down.
I'm not speaking to the just like,
Your left?
yeah.
We don't have
Right up front. Come to the center now.
There's something.
See the dead boomerang, and then there's a muscle behind it, right behind that. Stop, stop, stop.
That. Turn it up. Yep. Got it. Oh, look at those. They're so little. I didn't even see them. Oh. Okay, that's why. Oh my God. I didn't even know that's what they were. Okay, now we're fully zoomed in. I can't go any closer. Look at those, that little pile of them on the crack of the mussel. Oh, whoa. Okay. That's the mother lode right there. Okay. I don't know how deep even saw that. Huh. Right, so that's like a bunch of brittle stars hanging out in the mussel and on the mussel, and a little sea anemone and all sorts of snails and other critters. This is awesome. I think just let me know when you want to sample or slurp the brittle stars.
I think that's like, let's get the bathy first.
Okay.
That will play space.
Yep.
That we can then like just
Okay.
stir.
Yep. So we'll get a couple samples of the mussels first, and then focus
From,
on
like,
where we
around
collect
where the
the mussels. Yeah, around where we just saw those brittle stars. one of those, or several of those mussels, and we need 5 of the boomerang mussels.
Right.
These guys.
So you want that in a bio box, yeah?
Yeah. Bio box, right? Yeah. Yeah, I don't think they'd fit in. It's hard to tell, actually.
So 2B maybe?
We have, yeah, 2B sounds good.
Okay.
4, 25,
Okay.
295, 26. Is this 297? Is that right? 297. Okay. So we're gonna get 5 of the boomerang mussels.
Yeah. So you're picking up one.
Bathymodiolus. It's like a child.
And then boomerang, because it looks like a boomerang.
It looks like a boomerang. Okay. Hence the name. Okay. So we get 5 of those. What?
Diversity of questions.
Okay, so we are collecting some samples of these boomerang mussels and all the things attached to them. Then we are going to see what's behind them. And we've already seen when we zoomed in that there's lots of these brittle stars sort of lining the shells of the boomerang mussels, which you can't even tell that's what it is. If you're a keen biologist like Diva, you can see that. I had to zoom in to know what she was talking about. Okay.
Scammer is just like swim.
We did get one
That's weird.
earlier that had a
It's like
sponge on it.
This one.
So we're taking these as a platform for other things, but also sampling the mussels. And we've just begun to explore this seep, which again is formed by a really cool submarine landslide that exposed some of the methane that was trapped underneath, and we now have these bubbling sites that we've showed close-ups of earlier. We're now trying to make it to the head scarp where the landslide happened, just above where that slide happened. But before that, we're going to do some sampling. Shrimp getting out of the way. Look at all those brittle stars. Like tiny baby brittle stars. Are they baby or
No, I think
just their own little species of, okay. Wow. Yeah, we don't know when the landslide happened, but it had to have happened long enough ago that a community that thrives on methane had time to develop, and these carbonates had time to develop. We know there was one just from the map. I mean, this is landslide is a pretty obvious morphology, the way it looks, and you can see the scarp, head scarp, and you can see the chunk that just kind of moved downhill, and you can we saw that in the bathymetry data when we were mapping, because everywhere we go with the ship we have the multi-beam mapping. And so this was on our transit from one site to another. We had 2 potential seep sites that were located, and then when we looked at the data and cleaned it up, you could see that this was also a landslide. So that makes sense based on what we're seeing here. That landslide scraped as the motion of sediment downhill exposed some of that methane underneath. That provided an opportunity for that methane to reach the seafloor and the start of the development of these communities that thrive on these cold seeps. Yeah, check out the image. It's very cool. I mean, they look the same on land as they do at the seafloor. Just obviously, in this case, we're under 2.4 kilometers of water, almost. Cool.
Yeah, all right.
Hmm.
So you want it on B, right? So
Yeah,
right.
BB2B. And that's 297. Alright.
Up on your other page.
This is Bathymodiolus.
Okay, open that. Should drop in.
Can we just add the same muscle to the same
Yes.
bio box?
Yeah.
Okay.
Yeah.
So we need 4 more of those.
Yeah.
Yeah, so the cool thing about geology and biology is looking at the inside of these mussels and their shells, that the mineral that forms that opalescent, beautiful color is also calcium carbonate mineral called aragonite. So the active seep is also helping to ensure the structure of the communities that are living there. They have the ingredients that they need to thrive. So it's different calcium carbonate minerals, and different engineering, but everyone's benefiting from the methane that's coming out of this site. Yeah, so the outer part of the shell is made up of the calcite, and then the inner part of the shell is that aragonite. And that shimmering that you get, that sort of iridescent coloring, it actually is just like light diffracting off of the surface, which is why it often gets used in like jewelry. I think I have a few necklaces made out of aragonite. So one of the many impacts of climate change on deep ocean is on the ability of these mussels to create their shells. Does any changes in the water chemistry actually impact the ability to form minerals like aragonite, which are obviously essential for these mussels?
For anyone just joining us, this is live footage from a remotely operated vehicle named Sebastian. Currently working, it's like about 2,351 meters below the surface in the waters of Trinidad and Tobago. Found this amazing seep site today.
These all have the same number? Yep.
Where we have methane actually bubbling up out of the seabed and all these organisms that live on that.
So somebody asked when this slide happened. We could actually use the aragonite in the shells from the mussels and isotopes to try and isolate exactly how long ago that happened. But we do know that it was long enough in the past that it led to the development of this enormous mussel bed. And this is just a tiny part of it. We've just started exploring this area, and we haven't even gotten to where the landslide happened. So we're in the process of collecting some mussels. So biological sampling for a little bit here. And then we're going to continue and head up towards the scarp, which is the upper part of where the landslide happened. So right above where there is no disturbance, that's called the scarp. Cool, we got a couple of those brittle stars on this one. Almost got 2 for 1 there.
See those little things that it's like a little tiny strip of the colored. It's on a piece that's in that house. Just
This one?
yes.
Okay, so while we're here, going to talk a little bit about the aragonite again, because I think it's really cool.
Little colored.
So it's not just the methane that allows you to develop aragonite. You actually need this partnership with the microbes to make this happen. And this whole process is called anaerobic oxidation of methane. So you have these methane eating microbes and sulfate reducing bacteria that together team up to eat and consume this methane that's coming out, and the sulfate from the seawater, and that leads to a spike in alkalinity and biogenic bicarbonate, which is then precipitated into this calcium carbonate. So it's many steps and many ingredients necessary for development of these shells. We got, I think that was the, was that the fourth one or the third one? Fourth. Yep. I had four.
Okay.
They have four too.
So no más por favor.
So we're on, we need one more.
Two for one.
Two for.
Do you need 5?
Yes, take them.
2 for 1.
Oh. Wow. Look at this. Brittle stars hanging on there. They're trying
that is
not
like
to
a
be part of
cover.
this sample. Oh, yeah. Okay, if we're lucky, we'll get 2 here. Oh. We're gonna, okay.
He's in the.
Oh,
You'll get him.
he'll get him. Okay.
No, you'll get it when it comes on.
Yeah, he's on the porch.
Yeah.
All right, one's on the porch. The other one's going in the bio box. Okay. I don't know how to. I'll draw a boomerang mussel on the porch. This is part of 297. Is that okay for our 5th one? If it's on the porch? Okay. All right, we'll get him. Cool. Do you want to slurp up some brittle stars? Yep. Okay. So next up, all the little things. The stars, the little snails.
Let's throw that on first.
Stuff between the mussels. Yeah, yeah, of course. I'm not sure I can do that. Get to see all the glamour. That's why you have to do yoga. You just did. Yeah, so these, the aragonite on the shells is also, for geologists, used as a thermometer. So it can tell you exactly what the temperature of the seawater was, where it was formed.
Yeah. Push out all that. Alright, see if you can get the light on that. Let me try that. probably locked it through.
That's not clear.
Okay.
This is the same one as all of the
They have one.
above and below.
Okay. So if you're just joining us, we are on a new seep site, previously unexplored and unmapped. This was something that we, on the transit from site 3 to site 4, I believe, we were mapping the whole way because one of the objectives of all deep ocean work is to map the seafloor. And in doing so, we found a little indication of a potential seep, and also found that this is a submarine landslide site. So it's a good target for
Okay, coming to the round.
for potential seep site, and it is clearly an active methane seep that was likely exposed by that landslide whenever that happened. And provided a pathway for all that methane to make its way to the surface and get released to the seafloor. And this is now a thriving community of mussels and sponges and eel pouts and shrimp and all the chemosynthetic animals that thrive in these systems, and little brittle stars and amphipods and yeah, anemones, all the things.
Try not to, if you see any shells coming up, do an interception. So there's only get these smaller creatures. Yeah, so
Remember the slurp, the mountains?
Okay.
She wants this sponge.
Okay.
She will also be up here.
Okay.
Here.
Oh, Shirley's up here. Oh, good. Okay, perfect.
Perfect
Yep.
time.
Perfect. Yeah, if we can get into those brittle stars. Whoop. Don't fight it. There you go.
alright. So if you come down right there, there's some brittle stars right on
Yeah.
that. Just point at them so you can see.
Yeah. So like all these, or you might have to move that out of the way, but all of the little hairy looking things up there too.
Yep.
Oh,
Yeah. Fine. Do you want to get some of this substrate right there? There's like on the soil.
yeah, I mean, if there's snails also that we were interested in collecting. They're hard to see. Some of them may be on the shells as well. Yeah, the specific number of mussels is, some of them are for genetics, some of them are for pollutants, because these are filtering and concentrating some of the things humans have introduced. What else? We do with the, yeah, vouchers for the museum, so examples of the species. So it's not just a single, when we collect something, it's generally not for a single purpose. It's different studies that are aimed at investigating different things. As I mentioned, the mussel shells can also be used for looking into past climates, thousands of years ago or millions of years ago, depending on when they formed. These are more likely the recent past. They represent potentially a thermometer for the oceans in which they were formed, or the ocean conditions. I don't think these are the same shrimp as that we saw in the apartment complex on the mud mound.
Those were,
Those were, what were
those
they?
were amphipods.
Amphipods.
These
Yes,
are shrimps.
these are shrimps. Yeah. We didn't see much of them except for the few that came out of their little apartments, but yeah, these are chemosynthetic shrimp.
Should I keep going? Yeah, keep going. Let's stop. We'll go over the cloud right now. No,
Which
three.
suction sampler? 3. Okay. And this would be 298.
You wanna get one of those rooms again? Or that?
Which one?
Similar to what you guys.
Oh, yeah. Oh, yeah.
Can you zoom in to see what it is?
Yeah, can we zoom in on it? See what it is? Huh. Oh, it's another one of those little sea cucumbers.
So you don't want that.
I think we got
Yeah,
we got 6 of those. Yeah, we're good.
Alright.
Yep.
So you want seven?
I think we got yeah.
Six is good?
Yep.
Alright. 'Cause they cycled them.
Yeah, so earlier in this dive we saw a pile of sea cucumbers that were a larger or adult maybe form of the ones that we were just zoomed in on. That look like, yeah, little gummy worms. Okay, we were hoping to get some of the isopods or snails or other little things that are nestled in between the mussels, but
Right on top of this thing there is a.
oh yeah,
Just point that out. So
yeah,
see
you mean this
Yeah.
right here?
Yeah.
Yep, right there. That little guy.
Yeah.
Whoop. Go, go, go. He's in there. Okay. All right. I think you got him. No? Is he hanging on? Oh, he's hanging on.
You need to get the whole little spring. Yeah.
The whole thing. Knock it off there. Okay.
I'm not sure if we spoke about this already, but like this is a really good example of why the species and that models in general are ecosystem engineers. They're
You
interesting
see the
species
little
because
one
they
here?
hold like
You
substrate,
need to kind of scrape
like
off that little white.
shelters,
white
all these little animals live in. So
Who,
they're like
who is?
the mussels.
The mussels.
Yeah.
I did.
Correct.
I just, yep. You missed it, but I gave
Sorry.
a very boring speech on this earlier.
Yeah.
Yep. The engineers. It wasn't, but it was about aragonite and all the other, yeah. Yeah, so without the mussels, we wouldn't have any of these things, yeah. Okay, so I'm turning it over to world-renowned sponge expert, Shirley Pomponi. She's coming from Florida to take over the next hour of excitement on this dive because we have lots of crazy things to look at today. So we have We got here's 297. So in BioBox 2, we got 5 mussels.
All right.
Well, 4 in here and 1 I drew.
Yeah.
He's on the porch.
Oh, he's on the porch.
But
staying
he's there.
Yeah.
And
okay.
then we just slurped up some shrimp shrimp and brittle stars, as well as a couple of other things that I couldn't
And they're in
identify.
Okay, so they're in. Where are they?
Oh no, I wrote them in the wrong spot. Sorry.
Sorry,
This
those are Niskins.
Yeah, sorry. 298. Shrimp. Brittle star. Ignore this.
All right.
This is-
So
sorry.
those are in this suction sampler.
Yep. So
Okay.
3 is open still on the Niskin.
3 and 4 are both open, right?
No, 3, 5, and 6 are open. 1,
Okay.
2, and 4 were used.
For Niskins, for
Yep.
actual Niskins.
Yep.
No,
So, let me
1
see some.
and...
Oh, it didn't fire.
Yeah, it
Okay.
didn't fire, and 3 didn't fire, so there's only 2 and 4 we have this one.
Okay.
Fire.
2 and 4
So-
you have. Okay.
Did not fire. Did not fire.
Okay. Got it.
I'm just gonna write that.
All right.
So
Okay.
Yes.
before you start, we want to try to like scrap one of these limpets. or some of them.
Yeah, the ones that are on the
Yeah.
mussel shells.
But
Yeah.
I think you will need to like, scratch them with the suction
With the
samper and then
Yeah. Okay. We'll try. We'll try and do that. Okay.
Okay. Cool.
Okay. So, Sonny, see these little.
Okay.
That
Yeah.
those little. I'm not sure the best way. I don't know if you can suction it up and then drop it in.
We could try to do it, no, they all want to suck it up at the same time.
What?
Okay.
We have a little lip on the red thing,
Yeah,
so you
right.
use that to
Yeah.
kind of scrape things off.
Well, you won't be able to scrape it off. They're really tightly attached. I mean, you can try, but you can try. Yeah.
No, so long.
Yeah.
See if you could
Okay.
just scrape that little white thing
Okay.
off or down there. Suck it up at the same time. You may not be able to.
What?
You do
Hi,
it.
everybody. My name is Shirley Pomponi, sponge biologist from Harbor Branch Oceanographic Institute at Florida Atlantic University. Yes.
Up a little bit? No, no.
Okay. All right. If you've been following along this morning, it has been a pretty exciting dive. And if you're just joining us, stay tuned because it's going to be even more exciting. We've had, let's see, about 5 hours ago, I was asleep and my sponge radar woke me up. I woke up, turned on the video in my cabin, and sure enough, there was this big area of glass sponges, kind of glass castles forming a substrate for some other interesting animals. We saw some, what I think are probably ribbon worms. And we've been exploring this site for a while. It's pretty exciting because, I mean, our target for this dive, primary target, was to see if we could find seeps that Dr. Rachel Lahr and Dr. Rob Heron have been doing some mapping of this area, and the data that they got from multi-beam maps suggested that there were bubbles here, which would be an indication of these methane seeps, but you never know until you actually get down here and we can validate it. So I think it's particularly rewarding when you get down on the bottom and you see, yep, sure enough, the multi-beam data and the inference from that really showed the presence of these methane seeps, and the incredible community of mussels are supporting all kinds of life. So right now we're trying to get like a limpet-like organism that's attached to the mussel shells. I don't think we have any samples of those at all, and so we're collecting some of those. Immediately before this, I think we got some of these tiny brittle stars. There were some larger brittle stars that were collected. And if you look just kind of up at the, I'm going to say maybe 10 o'clock area of the screen, you can see sea cucumber. And earlier on the dive, it was like a Medusa-like arrangement of several of these sea cucumbers. It was really, really interesting. So if you weren't present for that part, you might want to rewind your YouTube video and take a look at it because it was really incredible. So this mussel bed really is a host to just a very diverse community of organisms. Hmm? It's not working. I think they got. I think some of them came off, but it would be
No, no,
maybe
it's
pick it
not.
up. Maybe just pick it up with a.
Yeah, maybe we can select one that has many. Let's see around.
It's just not. It's not allowing you to get a
Yeah,
section
we're
on
not.
it. Yeah.
So we're gonna grab
Maybe select. Maybe select one that has a few on it and try and use the claw to pick it up.
look for one, no, the one pick
Yeah.
up the actual full thing.
Yeah, that's good.
Yeah.
Back to yeah. Come up with that.
That one also has
This
an anemone.
Yeah.
Yeah, good.
Yeah. There are a lot of these really amorphous sponge masses, really common here. We'll pick a piece of that up later on, just to confirm whether it's the same species as one that we collected the other day. I think these are demo sponges, not hexactinellid sponges. I did a.
Okay, good release.
Yeah. You're gonna try and do the claw to get it, yeah. And then also, Sonny, after we've done that, just a piece of that sponge too.
Okay. Pick up. You want that one with two of them, or you want?
Yeah, because that's, the one with the two is fine, whichever one is gonna be easier for you to go in. One that has more would be better, because that only has the one, it has the.
Okay.
Let's see if there's one, yeah, if you can get, it's kind of buried though,
Yeah,
so,
we'll try.
let's see.
So we're going to the one with just two here.
Yeah, there's
Just
like
point it.
this one, but that's kind of buried. Is
Just
there
try it.
another one? You can try it. Yeah, if not, yeah. Yeah. If not,
Open it, draw. Open. Open. Down, down, down. Down, right. Close that. One can.
you wanna try, try another one? Should I have? Try another one?
Can we zoom in to see if they got
Bring
them?
it on this.
Yeah, when it comes up, we'll see how many are on there, yeah. All right, let's get
I
it.
want to see if it's have the animal. There, right?
So there's one,
And rotate. So there's two of them on.
there's two of them, I see.
Rotate the draw.
Let's see.
There's one
Yeah,
up
so
there.
there's one here,
One there.
one there.
Turn the draw again.
Okay, yeah, that's good, that's
Turn,
good.
turn,
You-
turn,
Yeah,
turn,
bring
turn.
it.
So there's
turn.
three of them.
Yeah, yeah, there's three, okay, yeah.
So the same buyer box.
Let me
I
see.
think it can go in the same one.
Yeah?
This one.
Yeah.
Okay, all right,
So same buyer box, right?
all right.
So bring it on, then you come up with the porch.
I think we
This is
can.
299, Sydney, is this 299? All right.
And there's one on the video. There's at least 4 on this one. Hold on, hold on, hold on. Up.
Up.
Right.
I think we will leave this as 297 and just put another one.
Okay.
Because then we will, all of them will
That'll
get
be the,
mixed.
okay, and then, but then are you gonna call 299?
No, it will be the same
We'll
one.
just, okay, so just gonna, all right, so we're not,
Just-
so it's gonna be still 297?
Yeah.
All right, all right, all right, and then we'll pick those off and they'll be the subsamples.
Subsamples.
Okay, got it, got it. Okay,
Okay.
Sandy, you got that, right? Do you want anything else that you look from here? I'd like to get a piece of that sponge just to confirm that it's the same one. I don't know, Sonia, I don't know if you need to move.
Hold on.
Okay,
Let's have a look.
great, thank you, good.
Page 2.
We have the Niskin as well, right?
Oh, yeah, do you wanna do, did we do one at the seat yet? Do we have a Niskin at the seat?
We did some, but I'm not sure.
Okay,
where.
well, let's do that. We wanna, so we'll get close to a sponge, we'll do a Niskin, water sample, and then collect
Okay.
a piece of the sponge, please. So one of the members of the science team Robert Cassidy is working on eDNA, environmental DNA. So and sponges are natural filters of what's in the water. And so what he's trying to determine is if these sponges in this area can be used as a kind of a proxy for animals that are living near or nearby or swimming past in this environment. So what we're going to do first is take a water sample, and then after that we will take a sample of the sponge, and then that way he'll be able to compare what he sees in the sponge with what he sees in the water sample. Bless you.
How big of a piece do you need? 'Cause this piece is loose.
Mm-hmm. Yeah, that's something like,
I think if you have a bigger piece. Yeah, so like this piece here is loose.
yeah, that would
You
be
want
good.
that?
Yeah, that's fine. But after you take the water sample.
Yeah.
Oh, and so if you missed the sea cucumbers this morning, you can see them. I think you can see them on the screen. I'll let you know where it's going into. Oh, wow, look at all those. Nice. Nice. Some nice view of the sea cucumbers.
Let's get some clams. This is the first time we've seen live clams.
Which one? Which one?
All
Oh,
of
the
white
that
live
one over
clams.
this.
If we could grab 6,
Is that what the one you want? Yes.
all of them. Yeah.
Those clams. Okay. All right.
This
So
is the first time we've seen them alive, Shirley.
yeah.
So let's
Okay.
grab 6.
Like these in here.
Yeah, there's loads.
Yeah. Okay. All right. So this is the first time we've seen seep clams alive. So we will, once we're finished, you let me know when we're finished with the Niskin.
There. I'm gonna fire now.
Okay.
No, I'm gonna use the salt drill.
So we're getting ready to take a water sample in this area.
Okay, so we're going to Niskin number 5, right?
Okay, it's open. But,
5.
yeah, 5. So then it's gonna go into 5.
So
Yeah.
besides the
Yeah.
sponge.
Sponge and then the clams.
And we would take a few more
Suction.
sea cucumbers.
Okay, all right, thank you. Okay, so we're gonna, what do we
So
want, the
this-
sponge?
Yeah, this piece right here.
That's fine. Sponge, and then after that, the clams, and then after that, some well, if you're, it's easy enough to get the sea cucumbers,
Yeah.
you can do. So we want sponge, sea cucumbers, clams,
Okay.
in whatever order works for you.
So let's go with where do you want what?
Well, the clams can go in suction, right? No? Sorry, not the clams, the sea cucumbers can go in
Yeah, they
suction.
have
Yeah,
number
they
2.
were tiny.
Yeah, we can do the sea cucumbers in suction.
Uh-huh.
The sponge, if there's space in the bio box, put that in the bio box.
No, the big one. This one available. Is that too big?
Oh, yeah, do you
I
wanna,
might save it for later. in case
yeah,
we go.
Could
get a
break a
small
piece.
enough piece to put in a quiver,
Yeah.
okay?
Okay.
All right,
All right. So Marcelo break that one. Break with the first, right? Yeah. So we're gonna break up each of this one. We're gonna break up each of this one. Small enough that for them to put right. So bring that one out.
299. Right? Right there?
There's one open already.
Yes, yeah.
The number one is open already. So the piece behind looks
Mhm.
like this and it says.
Mhm. Not too much.
Here, rotate the drum. Okay. That piece here, how should you go?
That's good. That's good. That's good.
So
Bring it in close so we can see it. Get
come
some
in,
good
get a
video.
picture of it. Or just go up to the dock. We'll stop a little bit. Sure, look at right order. So right thing up at the dock.
Mhm. Mhm. So I'm pretty sure this is a demo sponge, not a glass sponge on a hexactinellid. I've made some spicule preparations that I need to look at, hopefully later today. The spicules are the microscopic skeletal elements that are found in most sponges and help us to at least get to a general idea.
Okay.
Sometimes down, yeah, that's good. Sometimes down to the species level for these sponges. So I'll be comparing the spicules from this sponge with those from a similar one we collected a few days ago. We're hoping we're going to get this before it breaks off. Oh, I was afraid that was going to happen.
Okay, we could probably get it now.
Yeah.
Okay, put what you got in there. Okay, so that piece will fall off. It's right behind the shell. See it? Right behind. I need to let in.
It can honestly be any piece. They're all the same.
was the one that was already.
Yeah, yeah, you can just take a piece off of that one that was already cut if you want. Yep, you can get that one. Try not to, oh, it's gonna, same thing's gonna happen, I think, when you get it over to the,
Okay,
try,
to get it
okay,
as
yeah,
fast
yeah,
as you
yeah,
can.
okay, okay, so.
Pull up. Look. Pull up. That's alright, we were stuffing it. Gotta go very good, fellas. Okay, if you open your drawer there, it should fall. Yeah, that's enough.
Got it, okay.
So you keep it there.
So that's 300, is that sample number 300? Okay, and that's gonna go in quiver 1, yeah, yep.
So let's close these on. Come up first, close your drawer. Come up first, close your drawer and then push it back. Yeah, close your drawer there.
And I'm just pushing.
Right, okay, that's good. It'll fall in. Don't worry about what's in your drawer.
Yeah,
That's fine.
that's fine.
So just close it now. No. Yeah, close. Okay, push open. Close and okay. Okay, good.
Okay, good. I'm in good position to get the sea cucumbers next. You wanna do that next, or you wanna go for the clams?
Probably go for the clams.
Okay.
Where do you want to find?
Clams, you wanna try and
Yeah, I
pick them
think
up
they
with
are
with a
more-
claw? What do you
Yeah.
think? You try and
Yeah.
pick those up with a
So
claw?
you want it
How many you want?
quiver as well?
We want.
Six.
Yes, they can go in the quiver.
Okay.
Yeah.
So
this
open
one
number
six,
2.
7.
So
Seven,
when you
yeah,
take it
okay,
out, bring it
yeah.
to
See how it's
the
Yep,
right
yep,
side,
yep.
let it fall
I think
below.
We can,
They can maybe
they go,
in
in there, yeah,
this,
yeah,
but
he's gonna open number 3, right? Or whichever one. 2, okay, all right.
Running between 2 and 4. Raise it up a little. You can leave it on top there. It's fine. Once it doesn't fall over. Yeah. Here release. Okay, we're on 6 of the cloud.
7,
7. 6, 7.
6, 7. Sound like my grandson.
So, watch. In the yellow, but more to the end. More inside. Not just football. You want it? Go down. It's too hard, but just try it. Okay, kind of reach it. You want to crush it, but
So we're gonna try and get 7 of these. 5 will be for analysis for microbes, heavy metals, microplastics. 1 will be intact for a museum sample, and 1 will be used for other subsampling, so genetic barcoding, cryopreservation, so on.
Okay, try that. No. When you're not touching it. Okay. See how it works. So once you're happy, I'll try to take a picture of it. So stop right there. Stop.
Mhm.
Okay. Go for it. Okay, open right there. Right on. Okay, 6 more of those.
Okay.
So that's 301. There were 2. That's 1.
Which one are you going
6
for?
to go.
These there? Which one you want to go? Yes, behind the Joe or these right in front of you? Okay. Zoom in for you. Okay, hold it right up.
Can you turn on the lasers too, please?
Sure.
Okay, so we can get an idea. Lasers are 10 centimeters apart, so that gives us an idea of how large or how small this clam is.
Okay, open. Which one you going for?
Yeah. Want to make sure we get the ones with the hinges down into the sediment. What are we going after there? What's that? No, we don't.
There's 2 in the back.
Yeah, there's 3 back there. You think it would be easier to suction them?
Sometimes those hard stuff gets
All right, we'll
stuck
see,
in the,
we'll see.
Sometimes.
Yeah, okay. These are pretty fragile.
Okay, free data.
Mm-hmm.
You keep in count, right? Shirley? This is number 3. We got this on it.
Mm-hmm.
Open.
That's 3.
It's 2 over there.
Crushed.
There's 2 of them. Yeah, that's
Yeah, okay. We'll go ahead and do that next. What's this one? Oh, we can do it now. Yeah. Yeah. Cool. Cool.
okay, so that was probably too far.
Okay.
Up to these away.
So right now we're repositioning Sebastian so we can go in and collect 4 more clams. You can really see the diversity of fauna attached to and crawling around on the mussels. You see some beautiful tube worms
Amen.
and of course sponges. If you turn the laser so we can get an idea of the size of those.
Yeah, they're on.
They're on? I can't see
Yeah,
them.
right,
Okay, yeah.
see
Oh yeah, I see them. I see them. Okay. Thank you.
Go straight in that video.
Mm-hmm.
First one.
Come out with your own.
Yeah, we have some more blocks to collect. There was first, two blocks, and there are just one here.
It's 4. Mm-hmm. Yep. 3.
7.
Seven.
You're so cool.
We're moving Sebastian just a little bit closer so the manipulator arm is not so stretched out and make it a little bit easier to collect.
Bubblepalooza, man.
Okay.
It's a lot.
Yeah, that's because I'm sitting on it. It's because he's pressing it.
What are you doing that? Pushing. Pushing out that methane. That's something good at that.
Right, so what we're doing, we're collecting these
Clams.
clams. How many more we need to get?
3.
3 more.
Oh, weird.
So
Okay.
just picking it up these over. I'm putting it in the
Same
number
quiver.
But
two.
they're
Yeah.
really fragile. So.
Okay. They're just a tip. So
And then
Yeah.
after that, After that, we
You,
want
you,
to,
sorry,
go ahead.
You can't pick it up without breaking it, but if you just do a little extra,
It goes, it just,
and it's red blood that comes out, looks like.
Nice. Perfect.
Yeah.
and then after that, we want,
Some
sea
of
cu-
these, yeah.
sea cucumber. I think just one.
The same ones
How
like
many
we
sea
got?
Yeah.
cucumbers? One more sea cucumber, or just one this week? How many sea cucumbers?
To the left, there's a bunch
Whatever,
of them.
whatever we can
uh,
get. And then,
We've
what
got
else?
6. Is it the?
You got six
Clams,
this morning.
clam sea
Yeah.
cucumber.
Another 6 because the baby uses them.
Right.
Yeah, it's like
So
they
look,
seem to
look
be
there right
tiny.
there.
Yeah, so we got.
You can hear a little activity right now in mission control. We're just having a handover for the pilots.
No, we have 3 more clams to do.
Crazy, I tell you. No, I know. I can't do the island lean, man. Too much of a mainland. I mean, if we got a quiver open, we can go in there.
Yeah, okay.
So
We,
you're gonna
we
grab
do,
those
we have a
white
quiver
ones.
open, we've got 4 in already, we need 3 more.
3 more, okay. They say they're super delicate though. If you do break them, apparently they shoot red blood.
Yeah. Everybody will know.
Sorry, it's like those tube worms. You got a sample on yours.
So if you're just joining, we're on a beautiful methane seep, lots of bubbles coming up, and an incredible community growing in, on, and around these mussels. Right now we're collecting some clams.
So you see the all on the edges?
Yeah.
We've got either methane bubbles or hydrates starting to form
On the camera?
On the camera.
Oh, jeez. Oh, my God. Can they, they can't see that, right?
get hit with like a bunch of bubbles and then it looks like that. So
Can they see that on the live?
I could show it.
Oh, my God.
I think they have it now.
You got it
No.
now? No.
They've got that for you.
I know, that's the one I'm talking
So,
about. See all on
yeah.
the edges out?
Oh, I was talking
Yeah.
about this guy.
No,
That's your
well,
head cam.
no,
It was
we're,
pointed straight down into the
Zach
bubbles.
just noticed that you can see the methane ice, methane bubbles right now, methane bubbles on the
Oh, yeah.
camera cam.
Right.
I think we're going to switch the view so you can see it on the live stream.
Yeah.
Yeah. Collecting bubbles.
If you look up in the corners of your current view there, you can see the bubbles collecting. And then also the camera that's on the manipulator is currently pointed straight down. And it got hit with a bunch of bubbles and it looks like this now. So we might have some methane ice on that as well.
Can we just try suctioning up 3 of them? That one's, I think he squeezed that one.
Okay, that one's no good?
I don't think so. I don't know. We can look
Okay.
at it. We can look at it. Yeah, it looks like it's,
Yeah, see the.
yeah,
I see that,
no. Let's try suctioning up 3.
Okay, sure.
We've got 4 in the quiver. And
All right,
let's
you wanna grab
see
the
if we can get,
suction?
see if, yeah, okay, great. All right, so Sydney, we have 4 that are in coral quiver 2, and we're gonna try suctioning. I'll let you know into what bucket that'll go.
Jar of 4, okay?
That's fine.
Yeah, I'm interested to see what the manip camera looks like.
Marked?
Will be the same number. We're not gonna give it a new number. It'll be just the same number.
Okay, I gotta move to get you over there.
Sorry? No, you want also want ones also, so we're gonna put this.
After this, we're gonna get 3 of these, they're gonna go in here. And then we'll get the sea cucumbers.
Okay.
And then we'll kind of
So
get the
you're
ones.
trying to approach the secret.
Yes. Yeah, well, right now they're trying to approach he's getting us in a better position, so,
Okay.
Mike can get the, clams, and then we can then suction the sea cucumbers too. Sorry? Yep.
Yeah,
What?
Give them the same number, right?
for the
Sea cucumbers?
all of them, yes.
Yeah.
But those will be in a suction,
Yeah.
suction yard tank,
Yeah.
and then the one.
Yeah,
Yeah.
that's gonna be hard though, cause you gotta go through the sponge, cause you're coming from starboard to port, right?
Yeah, those look okay.
Okay. Very delicately balanced there. Trying to harm no muscles. Oh, rotate counter? Draw counter? Other way? Other counter? One more? There you go. That'll be a good angle for you. Which one you go for there? Okay, I'm gonna do half suction if you're ready. One,
2,
All right, lift it up and kind of shake it.
and
Lift it up out of there.
ah,
I think they got stuck on.
Yeah, there's two side by side.
We could drop them in a bio box maybe.
yeah,
You want to do that?
yeah, that's fine.
All right, so we push that. I gotta come up and smash muscles.
Put it in. We'll put it in this one.
Yes.
All right. And put it in 2B, bio box 2B.
Sub's gonna smell when it comes up.
Thank you.
Yeah.
Yeah, I'm gonna hold on. I'm gonna park up here. Look at all those smoke.
I know, dude. It's really gonna smell. I've been around when the methane push cores come up, and it's like, kind of smells like farts here.
There's Rachel's muscle.
All right, which bio box am I put in?
Let's put it in 2B.
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 That's the one all the way to the right, right? Yeah, 2B, all the way to the right.
Mm-hmm.
Okay, open in the lid. Alright, get over it, I'll feel suction. Killing suction, suction's killed.
If it goes in A, it's okay. Yeah, A or B. Either one will
Either one
work.
will go.
Either one works.
That makes it easy. Easier. One.
One.
Was that two? Oh, look, we got a shrimp in there.
Yeah.
All right. If you bring it out and look down the tube, point it towards the camera. Make sure it's not stuck in the top.
Yep, it's in there. Yeah.
There's one in there. So you got to kind of shake it a little more vigorously, I guess. You might be able to carefully tap it on the side of the box like that. Do that same motion, but just tap. No? Okay.
Straight that out.
Top of the box.
Alright, and last try. What about like this kind of action? A little bit. Going up and down. Doing that? Yeah. That is jammed. Alrighty. Guess you can't suction those guys. Copy.
Take this tank forward, please.
Stuck with anything there.
For oil and gas. Hopefully.
We really need to reverse on that thing. I guess we'll have to go back to picking them up. Closing tube. That's good. Another scoop. I can go ahead and just put that back, I guess. We're not gonna do that. You know what we should do, Mike? We should reprint the tip of that thing in TPU again, but make that opening as small as the smaller opening at the side.
Or don't blow nothing.
Yeah, so nothing can go and cannot fit.
Get stuck.
It's fragile enough you could poke it with Excalibur or something to clear it for a later section.
Yeah.
You won't get that clamp, but you'll at least have the hose again.
I mean, right now, I think it's a good sample.
If we can get it out.
Get it out.
If we can get it out later, yeah.
This won't go out of the tube.
Yeah, we'll have to get that out on deck, but it'll be a good sample then.
She has a question.
Okay.
Yeah, we're gonna leave it for right now. If we absolutely need the suction sampler, we could clear it by poking something else in there. But right now, that's a good sample for them. It's just unfortunately in the way. But we're gonna leave it in there, and when we get up to deck, we'll get the clam out and have a nicely sampled clam.
Okay, I've gone over the time I'm supposed to be sitting here. Just got wrapped up in the moment. And I'm going to hand off to Shauna now, and she'll be guiding you and our ROV team for the next 50 minutes or so. Okay, I'll see you tomorrow.
Hello. All right. Hi everyone, this is Shauna.
Gravel work, huh?
Mm-hmm.
Good? Okay.
No, yeah, it's one of the batimbrillus.
Okay.
The mussels.
Oh, muscle,
They're-
sorry.
The labels are better.
So you need two more of these, right? Two more?
Yes.
Yeah.
I think you want that one the shrimp's crawling on, and the other one the shrimp's crawling on. Or the one just below the shrimp.
I can see that the stars on the bow look really clear at night. It's amazing.
One.
We have
Think
some.
you can dump them in that quiver?
Some of the.
Today I'm just like.
Too large, you can spit it.
I'll send.
Force might get in your way, I don't know. I would kind of come straight, up, over, turn, and dump it in from the left to the right. You know what I mean? Yeah.
So after they finish this,
Mm-hmm.
there's also like some tiny gastropods, some brown gastropods.
Mm-hmm.
And then you start straight.
Can you see them
So
from
we
there?
can also scoop them, try to get rid of the sediment as much as you can, and then do the same for the sea cucumbers because the first ones we collected with the suction sample.
A lot more work.
Okay.
We scoop a bunch of them. We can put everything in the same biobox and then we sort it out later.
You said it's brown, what?
Yeah, once they zoom in, you will see them
Okay.
that it's covered by them.
Okay. And you want to know how many?
Then you'll be able to see it.
Let's see a scoop. Yeah. With scoop, you can like try to go through the surface. And
Okay.
we will have to do the same with the
Sea cucumbers?
Of course, we lost the suction.
Push forward.
Means stuck with the no.
Do we need to collect any more mussels or we got our mussels?
No, we already got the mussels. Can also use one of the firebox if that's easier.
You probably just scoop up right in that big cluster over there.
Like there's a bunch of gastropods.
Yeah.
Yeah. All of those.
Three.
See, it's full of them.
Yeah. Like these, yeah? So scoop all that?
Yeah.
If you're happy, I'm gonna torch watch out. Just make sure I can do it without smashing muscles.
You want flux?
Yes.
Probably will come up.
Let me just decrease the regard to the other side.
Mm-hmm. Mm-hmm.
Yeah, I gotta come up.
I think we can go for 2A.
Alright, I'm gonna float up, okay?
Oh no, there's no spot. Now we put everything here, including the signatures.
And then
And it's the dashboard. All that's it. So everything in there, we'll sort it out later.
okay.
Alright, I'm gonna push out.
And then 2 ones.
But for this, we need to start moving. Yeah. So, cucumbers. What's it called? Yes. All of that. Cucumbers, vegetables, and then just ones.
Okay. All in.
Everything can go. I mean, it's okay.
Did you do it? Is that top? Yeah.
Well, they share the lid. Whichever.
Okay. Just hold on to that for now. We don't know what we're gonna do next. It might be skewed.
We will need this good.
You need it again?
Yeah.
Okay. Yeah, just hang on to it. Just hang on to it. Scoop. We're gonna scoop it. Okay, what now?
Okay, we are interested in some small gastropods. We pick up this scoop and shake up some sediment. If we get back down,
Sure.
I could show you.
Okay.
As well as sea cucumbers. And there's some tube worms, like a large amount of tube worms.
So the sea cukes, just remember our suction is clogged,
Yeah.
so we would have to probably
With the same scoop.
Yeah.
either scoop or we could try to get Excalibur and like clean it out or something.
Or the
Since we need seeps, I think with the scoop we get a bunch at once.
with the cucumbers, yeah?
So maybe we can try
Okay.
it out.
Yeah,
first.
that's fine.
Those cukes right there?
Yes.
Yeah.
You wanna go ahead and do those, but we've got eyes on them, or?
Yeah.
And where are those gonna go?
Say BB1A.
One, oh, the big one.
Yeah, I think we're gonna put all.
Yeah, we can
Can't
do
you
that
drag this here?
together.
Oh, this is what it was. Okay.
Yeah, that
They ended up in here.
They went in there. Oh, okay.
I believe. Okay. Double check it with me.
And then Shana, when you land there, that it might also be covered by these gastropods that I showed you. So there's no need to go back to the I mean,
Is this all the same thing?
yeah, but the clams were in there, so in the back.
Mm.
So, but
Want
here,
to see that?
yeah, we'll be full of gastropods as well, so we can do everything.
So sea cucumbers, gastropods, but I'm not seeing any chickens. But we got those two first.
First the cutes because we have the cutes and the gastropods. Let's see if we see them. Yeah, they're gonna get the cooks first.
Mm-hmm.
So it's okay. Let them later. else.
Alright,
Those worms,
yep.
if them also come together.
That's okay. But we're interested in
If
these
it's on the way
cumbers.
and we get it. it's
Yeah,
okay.
that's fine. That's fine. Sorry. Oh, I was like, I knew I saw it there.
Están vacilando. You see them? The octopus?
I forgot to look. Do you see them? No, me though.
I think that would come from left to right.
I mean, is it tiny ones?
Yeah, I think so.
Seen some. Okay, around there.
Yeah, it's full of that. No, I'm trying to see. They will get it. Oh, we had a ship to shore this morning.
We are actually collecting some sea cucumbers.
Yeah, that's right.
Alright, where are these guys going?
1A.
1A.
Yes. I mean, it's a big one, but it's good color and things too.
You sure?
Yes.
Okay, enough. Do you want to go for another scoop?
We can try another scoop. See if we got the
Like
last
to the
one.
left, I don't want to run into your arm with the porch or pushing out, right?
Sydney, all of 301 went into 2A, yeah? That's a question. Did you see if they went into the other one? But those are all 301? All the same number?
301 is also in
You're gonna have to kind of flip that guy around where he's on our side. And end up with that. push towards you out there?
Mm-hmm. Weird.
Just shoulder right, and we'll bring them right close.
Mm-hmm.
So maybe you can do one more scoop.
Mm-hmm.
Before you, trying to like something else because it was full of heat, right?
Yeah.
Surface.
Yeah.
So try to do both. in the
Both?
same bite.
Two bones? Okay. And then 2 birds?
Yeah. Let's finish this.
Yeah.
Okay, what now?
Can we do one more scoop to get a few more of them? But there's also some tiny gastropods around at Point Bernal.
Okay.
And if we could get them both in one scoop and put them both in the same biowell, that'd be great.
Yeah, if you zoom in, you see them.
Yeah, quite small.
Like all
Go
of
ahead.
this. You see all of these tiny ones?
Oh,
The
little
brown
snails?
ones.
Yeah. The
Yeah,
brown
the
ones?
snails.
Okay. Oh, there's some right to
They're
the
all-
left side there?
Yeah,
Yeah.
they're everywhere.
Okay, yeah, okay. There's one right in the middle of
All
those,
hair.
right in the middle of the cucumbers.
Yes, exactly.
So if we could get a few,
Okay.
many,
So a couple more cucumbers and some gastropods.
whatever we could get.
Hmm?
Whatever we could get.
Yeah.
Sydney, those sea cucumbers 3 or 2, yeah? That's a question. Yeah? That was 2? I think it was 2. And then we'll see how much we can pick up now.
It's
And you keep
snails.
them out of the same
Small snails. I would just get the cutes first.
assessment number, yeah? Sample number. We're also
It's
trying to
like,
pick up a couple of tiny gastropods, tiny brown
See the
snails.
snails there?
So, keep an eye on it.
Those things.
Same? 3 or 1?
Before I move to
Everything
the
is
south,
Okay.
Zach, maybe we get these notes because I have plenty view. Because if I move, they disappear, they're very small. I can catch them all together.
It's like a rock or something.
Underneath?
Pushing a lot. Get some?
No. It's on the rock.
I mean, there's, I guess that's rock beneath. But
Or for the corners.
For the what? Cucumbers? Yeah, I guess so, because I think we might have to unclog back over. Get real close to the camera. Stop. Stop. Go left. I think we got to unplug the suction sample if we're going to get these guys. Sorry. All right, so you go ahead and put the scoop back. And we can get the Excalibur. We can get them out that way. You want to do that?
Yeah.
I don't think you're
You
going
mean
to
for the sea cucumbers or the
both.
both? Okay.
All right, I'm pushing out. Put that guy away. Well, do you have something in there right now?
No.
Yeah, so just put it in the holder.
On the spot.
How many more did we get? Two more? Two sea cucumbers, right? No, but in the last school,
Yeah,
there were two.
You had ones from before. Okay.
We should move to the next place.
Tobago?
Yeah, let's go.
Okay. Mike, I think we want to move on to the next three points
Oh, okay.
instead. Yeah.
Okay. So don't try and unclog it.
Yeah, at least not right now.
Sorry?
He's asking to don't bother trying to unplug it. At least not right now.
Not for now.
Yeah.
I think we already got some. I mean, some of the snails might be some of the suction samples, the mosses.
Do you want them to hold on to that then? No, let's just put it back. You're almost there.
Yeah.
Just go ahead and put it away. No, we're gonna go on.
You haven't done any transects, right?
Sorry?
You haven't done any transects. What's the question?
Maybe on the next slide, right?
After two?
Yeah.
So you're moving
I'm gonna
towards
see if they
3.2?
hit one. I'm gonna check if they already hit one.
And we're just heading towards 3.2, yeah?
Yeah. Just put the lasers on.
Yep, lasers are on. I think I just got to look up.
And we're not deviating unless we find something
Yes.
crazy.
Yeah, exactly.
Thank you. I know.
Well,
Crazy.
it's impressive.
We're gonna, so zoom out on your map. We're gonna go to waypoint 2 now. Towards waypoint 2. Yeah, basically, up or down there. Down the blue line. I don't know if we'll go all the way to the line. I'll probably just stay where it's most interesting unless you disagree. I don't think you're necessarily interested in the line. You're more interested in the seep.
Yeah, that's right.
I was
She
just
wants to know
saying,
if we want to travel exactly along the line until we point to
I think you're more interested in the seep, right? So just follow the
Yeah, so
interesting
let's
stuff.
follow the, like going towards the next one, but following.
Yeah.
Yep. Yep.
Oh!
Fall parallel to the line behind us.
Just leave them to the north of us, but so that I'm kind of on the good side, you know what I mean?
Okay.
And then just have them follow. A lot of interesting stuff on the sonar here. Look at that
Mm-hmm.
big hole over there. See that?
Mm-hmm.
Might cruise by there. That stuff on my camera, I'm pretty sure is high grade. I do not think
What are you, Jeremy?
bubbles. Interesting. It's not going. It's not moving when I'm
Yeah.
moving my camera.
go ahead. We're gonna start to follow south. If you can follow us up towards the waypoint.
Yeah, sure, no problem.
Thanks, have fun.
On chance.
How are you?
Very good.
Welcome to the paradise.
Jimmy's running the clock. Every morning, 9 o'clock.
Where you want to see?
This again.
really good. That is like a yellow.
Where are you seeing it yellow?
Like just like an overlay in terms of the color.
Like greenish.
Yeah.
Oh, I think it's just the distance from stuff.
Okay.
The water distance.
Yeah.
Get a little closer if you want.
Yes.
You want to get
Uh,
a
When we get there. Yeah, I think it's just the distance from the
Yeah, but you have to stay kind of back. There's like stuff sticking out.
Mm-hmm. Mm-hmm.
On top of this sponges and amazing. Yeah, both are sponges. Wow, it's huge.
Mm-hmm. Wow.
It just goes on and on and on and on and on.
Yep.
As you can see, there's more to the right on the sonar too. You see that? You doing there, man?
Yeah, I'm good.
Good sleep?
Good sleep. Good workout this
Mhm.
morning.
Yeah.
I was up at 8:30.
Oh wow, it's right through. It's been normal.
The last week and a half, just Rafa's been training me. It's been good.
Rafa's been training you?
Yeah, look at it.
If you want to continue on, so we want to. The same more bubbles, but
Yeah, that's what we do.
yeah.
Yeah, there are some coming out of there, see 'em?
Yeah.
Not so many.
I think I watched all of this dive in my room when I woke up. It's because I woke up, the TV's on, I'm watching it.
Mm-hmm.
This has been an exciting day for sure. This field is massive.
Was this here as soon as we reached the bottom?
No, but soon after. Some topography, about here. Go that way a little bit. Yep, there it goes.
Wow.
which one I had to see?
Continue on to.
On to two? And undertake, can you see more?
Yeah.
Oh, we're far from you. Oh. Roger. Kilometer. Trucking them. Oh, if they can speed up a little bit, that'd be good.
Aliens are.
Yep, I told you.
Bridge, Charlie.
That's where the saucer was.
Yeah, go ahead.
Morning, Rafa. What's the ship speed at the moment?
1.4 knots.
Can we come 0.6, please?
Yeah, sure, no problem. Good morning, man.
Good morning, mate.
Josh, have a reach? Sure, I'll go eat. We're trying to make it over to waypoint 2. Which is... All right. We just sped the boat out, but if it goes too fast, of course, just grab it. We'll see. Enjoy.
Let me just double check the core for the 2 keys.
Good?
Yep.
Close it?
Yeah, I believe so.
Is there something in it?
Yeah. 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 Some plans.
Bridge RV. Sorry, Rafa. Can you pause the minute, please?
Pausing the move now.
Continue?
Yeah.
Okay, Bridge RV. Disregard that, Rafa. Just continue to move.
Continue the move.
It's gonna close, right?
Yep.
Mm-hmm.
Zoom.
Put this camera down.
This one on.
Yeah.
two rooms. There is a clam stuck in the suction. So you know about, yeah. Exactly, and then get to the waypoint and we'll stop doing this. Okay, perfect. And I don't think we've done any charms. I think loads of stuff.
Okay, great.
I mean, this was
very
very
full already.
yeah. Perfect. He was suggesting when you're picking up these basketballs and two rooms, just put them all in.
Got it.
Thanks.
Howdy. So checking, then we're going to waypoint 3.
Three?
3.
It's not you?
No. So
So you want to cover the corner?
So we actually, yeah.
No
So we're gonna cut. So we're literally gonna run along this scarp because we're low on time. We're gonna just cut across and get to the scarp feature because that's what is geologically interesting. Please. Hello, everybody. My name is Dr. Diva Ama, and I'm the chief scientist of the expedition. I'm going to be taking over for the next hour from Shauna. We have been having an extraordinary dive. This is far beyond what we had expected and hoped for. So just as a little bit of background, we several days ago we mapped this area and we did pick up some bubble streams coming from the seafloor. Bubble streams are usually indicative, at least where we are, of methane potentially coming out of the seafloor. And we know methane powers these ecosystems, methane seep ecosystems or cold seep ecosystems. The bubble streams didn't seem to be that strong from what we could see from the mapping. So we had pretty low expectations coming into this dive because, of course, like mapping is absolutely the first step of what we do, but it's really getting eyes on the seafloor that is able to really confirm what is down here. We did touch down and started exploring, and within a pretty short amount of time, we did find these extraordinary cold seeps. Mussel beds as far as the eye could see, interspersed with tube worms, snails, brittle stars, sea cucumbers, lots and lots of different, at least 2 species of sponges, gastropods, etc., etc., etc. A lot of different animals all in this one small area. Methane seeps are incredible, like oases of life on the deep sea floor. You can see now, once we've moved off of where the bubbles were found, how different it is in just a few feet. So right now what we're doing, this feature that we're diving on is what we think is a sort of like a landslide under the water. So a slump where a piece of the seafloor has actually slid downhill. And so now because we've spent so much time at the seeps and done a lot of sampling there and a lot of getting video, we're now going to cut out one of our waypoints, which would have taken us onto the landslide, and actually head straight to where it looks like the landslide has detached from the seafloor above it. And so we want to have a look at that scarp feature, that sort of like what we think may be like a cliff face, to see if it gives any insights. We also, what time is it? We also have, in the remaining 3 hours we have on the seafloor, we're going to try to do that. And we also are going to try to do a heat flow measurement, just because this area is very interesting geologically. And then we're also going to try to get some push cores, time dependent. If we're able to do all of this, we will also plan to head back to the seeps, just because they were so incredible. Who wouldn't want to spend more time there? But this is a site that before today no one had ever seen or knew existed. So everyone who's watching, we are amongst the first people on the planet. We are the first people on the planet to see these incredible sights. And that's something that's so special and so humbling to be a part of. This is great, guys. Just keep going like this to waypoint 3. And so now that we've moved away from where the methane is coming out of the seafloor and providing that energy source to these seep ecosystems, we can see that we've reverted back to this sedimented environment. It does look like there's a bit more life than perhaps what we were seeing in other sedimented environments. We can see brittle stars, we can see shells of chemosynthetic mussels and clams dotted in between. There's some sargassum dotted on the seafloor. And we can also see how many marks and tracks there are, which indicate that there is a lot of animals around here that are, really bioturbating. So essentially burying and feeding and doing lots of things to shuffle around the sediment. Are you taking back over? Taking back, are you here for the next hour? So I was just saying what we're doing is we're bypassing waypoint 2. And we're just going to head straight to waypoint 3 because we want to get to that scarp.
Eva, you are a rebel.
You know, I call the shots on here, Zach. So
Some horses can't.
I can, exactly. Except for the captain. Don't tell the captain. So for instance, this track right in front of us that we can see that looks under the seafloor, that might be a sea urchin that is burrowing its way along the seafloor. And so all of these marks, which are called Lebensspuren, they are clues as to what life lives in these more sedimented ecosystems.
You didn't want to that break?
I have meetings from 1 to 3.
Oh, okay.
So I figure I better do my part now.
Well, we can cover that. I don't want to do an extra hour. That's just good for me. Alright, I don't want to.
I'll do that. You'll get your time. It might be more interesting. Oh, wait a minute though.
What's up? And so let's like deviate a little bit. That's what we're doing. We're exploring on the way to waypoint 3. We knew that there might be other features around. Before we found the seeps, actually, we did find what we think is an asphalt seep. So that's what happens when essentially like bitumen comes out of the seafloor and then hardens. So it comes out in a semi-liquid state. Because it meets this very cold seawater, it hardens, and it actually looks very lava-esque. And so when it does harden, it forms this like mound-like structure that lots of animals are able to go and live on and actually put themselves higher up into the current so that they can get access to more food. So we saw a lot of sponges, we saw a lot of brisingid sea stars, which are all filter feeders. Yeah, it's been that long. We're just in a time warp.
Isn't that exactly what you wanted for Christmas?
Time warp or an asphalt seep?
Asphalt seep.
I did actually say but I wanted to see when it was liquid.
It must be good.
So always that. So while we're on the way to the scarp feature, we do have the ROV sonar on, and that allows us to detect any structures or any bubbles ahead of us. Both are often indicative of different habitats and therefore different life. And so we do have something popping up on the sonar in front of us. So we are going to head over to check that out. And gathering, I mean, the whole point of this expedition, the Deep Wonders of Trinidad and Tobago expedition, is about gathering that baseline knowledge of the deep waters of Trinidad and Tobago so that we can use it to try to inform management and stewardship, essentially. So we know that one of the first steps of effective stewardship is knowing and understanding what is there. And then once you're able to do that, then you can that often leads to a growth in how much you appreciate and value these ecosystems. So this entire expedition is about bringing gathering baseline knowledge so that we can make better decisions about how to use the ocean here. Oh, great, some amazing rocks in front of us. And then also, it's about sharing this with as many people as possible, both during the expedition and after. So it looks like we're at another feature.
This looks different.
Yeah, let's see if we can see the top. Man, this is the dive that just keeps giving.
Wow. We're going to get big corals. That is completely different than just a couple of 100 meters away.
So I am not a geologist, disclaimer. These may be carbonates, old carbonates, and carbonates are often found in and around methane seeps. And so what we found in Trinidad and Tobago in the past, though it's often on a much smaller scale, is we get this zonation that happens in methane seeps. Right next to the flow, there will be mussels, the Bathymodiolus mussels we've been seeing. Then there will also, just outside of that, be tube worms, for instance, and other small animals. And then outside of that, then there often is this sort of halo of carbonate structures where there isn't as much flow coming out of the seafloor, seepage, but you do get these structures that allow animals like these corals to find a home and actually project themselves into the seafloor. Wow, it keeps going. Okay.
Very standing edge. Can you move the vessel a little more north? Just a wee bit. Wee touch. Okay. Wow, that's weird looking rock.
Yeah, so it does look lava-y, right? So then this may also be an asphalt seep as well. The structures in the rocks are very strange.
Are those shells?
These are all shells. So maybe it is then carbonate. But those are all, what we can see here, these are what looks like mussel shells or clam shells, maybe a mixture of both. And you can see that they look quite old, almost like they've been cemented into the rocks. Wow, 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 wow. Look at all these corals and anemones on the corals.
You want to go anywhere else after this?
We're trying to get to the scarp.
I'll just stay here.
And all our coral quivers are full anyway. Nearly.
I'll just
We have
stay
two
here the
more.
rest of the cruise.
Can we zoom in on this pink stuff? This might be bubblegum coral, which we actually haven't seen yet for this expedition. I think it is. And you can see there's still potentially some flow happening around. This is a very large tube worm off to the left.
You're right, huh?
How hard would it be to collect this?
Oh, I think we're good.
Okay. I know we're on a mission, but either we drop a waypoint here and then we come back to collect it after we get to the scarp, or we just collect it a piece now.
That's not going to happen. got the hook.
Okay. I appreciate your guidance and honesty.
You're gonna find something else.
So we can see here this perfect example of animals finding habitats that work for them. So these corals are using these hard structures to project themselves up into the water column so that they have access to faster currents. Because once you get close to the seafloor, that's where currents slow down because of the friction. So here they're able to move past that. And this is actually these are the first bubblegum corals we've seen on this expedition so far. We've also seen it right in this little patch, some bamboo corals. This one on the right looks like a chrysogorgid. So a diversity of deepwater coral families.
Really big coral stem there that goes off.
Yeah, and so we can also see Zach, the pilot, is sharing. We can also see in between we can see some stems in between that look very old. And so these might be fossilized coral pieces that have been here for a long time, old corals that were here once upon a time and then broke and then became fossilized. So it gives us clues as to how long these features have been here and how long life has been colonizing them. So,
Where do you like this?
coral quiver 4 or 3,
4 or 3, okay?
please. So funny, just the other day we were like, oh, we haven't seen any bubblegum coral yet.
Now you should say we haven't seen any aliens yet. There have been signs.
They have been signs.
Nice one, man. How much of this you want? Just one little branch snip?
Yeah, so we're gonna need a piece for genetics, which will be quite small. We want a piece for a voucher specimen, so we can look at its anatomy, and then they may also want a piece for cryo. So I think if you could maybe this like lower
That lower bit?
chunk.
Okay.
I'm not sure how. Can we just flick the lasers on for a second,
Sure.
so we can get a sense of scale?
Yeah,
Oh.
I think they're blocked by.
Oh yeah, here. They're right here.
Yeah?
So I think if we can get a piece like this big, 20.
Okay. So like just big enough to fit in the 12 quarter.
Yeah.
So that whole branch probably.
Yes, that'd be great.
Okay.
If you think that can fit, yes.
The only thing I don't know, man, is I don't know how fragile this stuff is. If you touch it, it might crumble, but all you can do is try, right? Just try not to bump it until you're ready to grab.
And interestingly, I know you have your hands full. Just below, it looks like there's a carnivorous sponge also, which we might want to get after.
Lurking.
Yeah. Because that is one of Shirley Pomponi's, who is on board, one of her specialities. Megan, what's your sponge speciality? Do you have a favorite? Yeah. What? That sounds like it's not even real. Oh, like from the Caribbean?
Yes.
Okay.
Purple.
The purple one.
That's not brittle, that's good.
Yeah, it's
That's a
brown.
good thing. It's like that white one the other day, that one. Oh, get it?
Yeah. Perfect. Just go with it.
See what we get.
Oh,
Get it
oh
off.
no.
Hey, look at this. That white stuff the other day, you just look at it, it'll fall apart.
Yeah, if you sample like Madrepora, it's like literally like China. And it just breaks, like, yeah.
Have any of you spongologists ever worked with Paleozoa?
I am not a spongologist, so that question should not be directed to me.
Super duper poisonous Hawaiian death sponge?
Is this a shallow water thing?
Yeah. always thought that one's pretty interesting.
Wow, look how can we just get a quick zoom of this before you pop it in the... Oh, beautiful. Look at that, how gorgeous.
So where's the polyps?
So the polyps have actually retracted. So where each of the nubbins are, that's where the polyps, because we've disturbed it, they've all pulled in.
That's bigger than it looks, really, you know?
Yeah. I have not showered today, Marka. Just so you know. Great.
Hey, Monica, while you're back there, somebody knocked my monitor all wackadoo. I'm then fixing that.
I will fix it for you. It wasn't me that knocked it, right? Was it you?
He's like, somebody.
I'm not saying it was you, but nobody else did that.
Is this Paragorgid?
And say so.
That's what I would say as well, but it's the right color. Knock that bit in, knock that bit in.
Might have to re-grab the tip of that. So it doesn't better fall in the way.
Yeah.
So always turn to where you can see both times. Also, it's really hard to do that.
So the Latin name, the Latin family of what we think this is a bubblegum coral. That's obviously the common name, and it's nicknamed that because of the color. But the Latin family name is Paragorgidae.
I was thinking ahead, you made that into 3 samples.
Yeah, exactly. We don't even have to cut it up now. But we'll get the other piece, right? The piece we knocked off. As well. It's knocked off.
Take that whole rock.
Nope. Do not want that. So this is what number? Well done, Manny. Great.
You just gotta do it again.
We did knock off a piece, so we would that piece hanging down. So it'd be nice just to grab some of that while we're here as well. And so again, this is a great moment to look on the right of the screen. You can see all of these stalks, coral stalks, very thick ones that probably would have been very old corals that have been here once upon a time, died, collapsed, been broken up, and been left here to fossilize. And in the deep sea, not at methane seeps, in the rest of the deep sea, life tends to be very slow. And so it means that a lot of animals can live to, they have very slow metabolisms, they live to very great ages, they grow slowly, they move slowly, they reproduce infrequently. And for a lot of them, we also just have no idea, but that's what we suspect because of the fact that there just isn't that much food, because of how cold it is, lots of different things. And there have been corals aged in the deep sea that are thousands of years old. Individual corals, for instance, gold corals that have been aged at, I think, around 2,000 years old. There have been black corals that have been aged at over 4,000 years old. I mean, I'm always blown away when I think about this. Like, think about all that has happened in human history in 5,000 years. That would take us back to around the time of the pyramids, for instance, or maybe even older than that. And so it's incredible to think that there are animals in the deep sea that have been alive for that length of time, and all that they would have witnessed in the deep ocean during their time. It just really shows about how different timescales of life are in the deep ocean, and why we do really need to tread very carefully in these ecosystems, because the timescales are different to what we are used to, and it can take an instant to destroy something, but for it to grow back will take thousands of years.
I think that's fossil coral.
Mm-hmm. Yeah.
Sure you don't remember this?
I don't know what to do with it. You can see the section in Egypt. You would put it on your shelf. We are, what's this?
It's one I made out of jasper.
Wow. Beautiful. You made that?
Oh, I made picture
Wow.
jasper.
Hidden talents.
Yeah, I keep all my talents hidden. It's very easy. There's not very many.
Oh man.
Look, man, you already got that thing in there. BS. It's in there? Yep. All right.
Great.
You want more?
Nah,
That's it?
that's great. Thanks, guys.
That's it for you. That's it.
Yeah, I think we got some pieces. Let's just head out. Head out to waypoint 3.
Roger. Rod.
Yeah. I need to stop being distracted by all the beautiful things.
That's a good problem to have.
Okay.
What are we expecting to have?
So this 3, if you look up here, Zach, is this, which is, we are here, and so this is a big slide, like landslide. And so this is where it's broken away from the seafloor. So the geologists are very interested in seeing that face,
Okay.
what this looks like.
Okay. So like the steep part.
Correct. Like the actual scarp is what we'd call it.
You okay if I come off while you're doing that? Yeah, go.
seep, right?
Potentially, yeah. So Rachel was saying at one of the other seeps we were diving at earlier that a lot of the stuff we were coming across were ancient seeps, which is just like these massive mounds of. So what this could be, and again, caveating all of this by saying I am not a geologist, I am a biologist, is we had heard from Rachel Lauer, who's with us from the University of Calgary, that these types of mounds are just massive ancient seeps, essentially areas where carbonate rock has built up and it's gotten all of these mussels and clams in the midst of it that become all jumbled up and then form these structures for different animals to grow on. Great. Let's like get over the top of this and then waypoint 3. See what else we see here.
No, that is condensation is inside of the lens.
That's water.
Yes.
Wow, it looks very viscous.
Yeah, which we were going to switch when we had a lot of deck time. The thing we did a test of the spare and it was not working. So next time we have any time on deck, we'll probably try and switch that with another one like this. So it'll be 2 4Ks.
Right, got ya. Just interesting.
Yeah, it's a shenanigan filled trade. Hijinks too.
We're keeping you on your toes.
With both. All right. You good,
Great.
Annie? Yeah.
Let's go. What's this one? A Caligorgia. You're like, good.
All right, moving on.
So we're seeing here a lot of coral species that we haven't yet seen on this expedition. This could be another Paragorgia or maybe a Caligorgia based on the color. The one that we're coming up, this is a bamboo that's just come into field. I have no, oh, that's a net? Oh, that's so interesting. Can we have a quick zoom here? Just a quick zoom.
Sorry, the lead said we gotta go.
Just super quick.
You see the net, right?
No, where?
Is that a fishing net?
Yeah, it must be, right? What else is it going to be? So this could be ghost fishing gear. Yeah. What else is it going to be? Okay, that's great. And then on the right, we can see a really big bamboo coral. Some squat lobsters in between, another bubble gum. I also feel like this mound is not insignificant. This looks quite tall.
Something important happened here.
Once upon a time in geological history.
It's quite tall.
So the white animals that we're seeing dotted around, these are not crabs, they're actually squat lobsters, so relatives of crabs. They're probably from the genus Munidopsis, very common deep sea inhabitant across multiple different depths, multiple different habitats. Oh, wow. And so we can see where some of these stalks have come from, right? Like that would be these types of corals we're seeing further upfield that are very large. So we can see the lasers on the screen. Those are 10 centimeters apart. So this coral is at least sort of 1.5 meters, 2 meters. The fact that it's a bamboo coral, look at how thick its base is. This, again, as we were saying, could be one of these very old corals. And we have very few of deep sea corals that have ever been aged. Wow, amazing. And you can actually see the bamboo structures where it gets its name from in those sort of horizontal stripes along the body that resemble the shape of bamboos.
There's another one up to my right.
And just for everyone's awareness, I mean, we are currently at 2,360 meters, so over 2 kilometers down in the deep sea. It just is always really extraordinary that we're able to get these images in near real time, and you can watch from your home or wherever you might be at this moment. Yeah, a lot of these big corals. Yeah, right? Yeah. Lots of old stems here. Mm-hmm. And that's what we hope for. We set out when we designed this expedition, we really wanted to get a taste for as many of the different ecosystems in our deep waters. And so we've done continental slopes, which have been very sedimented in the north. We've done the Barbados accretionary prism, where we are now, which is a lot of these seep-like environments. We then headed down into the abyssal plain, so around 4 kilometers deep. Then we went out into international waters to look at the connections with Trinidad and Tobago's waters. And now we're back on the Barbados accretionary prism, and we're going to be heading to a different part of the prism in a few days, where we expect to find some mud volcanoes, which should be a bit different. And then we're going to be heading into shallower dives, actually mesophotic dives, so much shallower, off of Tobago in a UNESCO Man in the Biosphere Reserve to close us off. So really there has been a bit of everything, I hope, and all of this information will really help us in the years to come to understand and potentially protect these places.
Am I on the right path here?
So I guess we are on the scarp, right?
What's that?
We are on the scarp.
Yes.
Yeah.
Yeah, we're kind of on the edge and in.
Yeah. Text the geologists.
Is that a giant mussel shell?
Yeah. So that's a Bathymariola shell. How did that even get up here?
No, seagulls.
And so we joke seagulls, but actually there will be some deep sea equivalent that, sure, of course, isn't a bird, but some deep sea predator that will have potentially picked up a seep mussel where all the seep mussels are, and then brought it away from the seep to be able to eat it in a safe place, perhaps. So where we are right now is called the Barbados accretionary prism. A lot of Trinidad and Tobago's deep sea is actually on this prism, a gorgeous anemone in front of us. And what that prism means is that it's essentially where the South American plate is subducting below the Caribbean plate. And when it does that, it scrapes off kilometers of sediment and crust, and that gets jumbled up onto to form this prism at the intersection where those plates are subducting one below the other. And that jumbling of material is what leads to jumbling and subducting, et cetera, of material is what leads to the production of oil and gas in the area, which actually powers Trinidad and Tobago's economy and has done so for decades. But it also leads to these incredible geological features and ecosystems that we are seeing down here in the depths. And the Barbados accretionary prism is actually the largest of its kind in the world. It extends from south of Trinidad and Tobago all the way up the Caribbean chain, nearly to all the way to Antigua and Barbuda. Hello. Is this the scarp?
Yeah.
Yeah.
Scarp. Yeah.
So all these rocks.
So we
just
So,
got
yeah.
on it.
So maybe this mound is what we just climbed, right?
Yep.
And then now we're starting to go in here. So then it should be on our right.
The scarp is on the right. Yeah.
So do you think we'll see something?
Not sure.
Do you
I
wanna-
think these are probably like the seep has migrated potentially from where the scarp happened to where we were. I don't know.
Okay, we're coming to more things ahead of us. It looks like the slope is increasing quite a bit here, no?
Yeah.
Am I imagining that?
We're going down actually.
Down.
Okay, so we're going down into the scarp then.
Yeah.
Getting more scarpified.
Scarpy. Yeah, it's right. We're on a slope, I think. Right?
Yeah, so it looks like we're going down into this now.
Mm-hmm.
Yes.
That was the mound we
Yep.
just climbed.
Yep. Yeah, so
Wow, look at these
everything to the left
corals.
is this- yeah.
Look at that. Let's just get a quick, let's pass close to it, close to its presence.
Smell it.
And so I always think, even though corals are animals, this is like the equivalent of, you know, the giant redwoods in California or sycamore trees or some very, very old tree that's been here for a long time. These corals are also similarly very old. I mean, this one again is even bigger than the last. And it provides habitat for other animals as well, just like trees do. Cool, let's keep going. Amazing. So it's a big bamboo coral.
Wide berth. Another one.
Wow. Man.
This is a new site, right?
Yep.
All of this.
No one has ever dived here before. We are literally the first people seeing this seafloor.
Surely not the last.
And big crinoid in front of this one. Definitely not the last. So what do you think these are, Rach? You want to jump on the hot seat?
Sure.
Or you want us to just spend some time like trying to work out what it is? Do you know?
No, I mean these are all carbonates.
So do you think they were? So do you think they're like in the sediment, and then when this fell away, they were all
No.
exposed?
It's hard. It's a chicken egg problem, kind of, because the slide.
More
would have
seep mussels
Yeah. So this
and
would have
clams.
when this moved, it
Yeah.
would have exposed all of this that was under, all
Sediment.
Yeah, lots of sediment.
Yeah.
So that is likely a lot of methane hydrate that was then exposed to the open ocean and started the bubbling. You would have then had formation of those giant carbonatey things. But
Mhm.
we also, at the same elevation over here, saw evidence of oil like seeping at the seafloor. So,
Yeah.
this whole slope here is connected to the hydrocarbon reservoir, and it's leaking both methane and what we were calling, what did you call it? Asphalt?
Asphalt.
Yeah.
Seepage.
Canadians say asphalt, and it really bothers me, but I say asphalt.
Well, remember that.
Yeah. Asphalt. That's not how it's spelled. I just will not let it go.
Aluminum.
Yeah. Aluminium, same thing. But yeah, it's not clear if that slump and landslide, submarine landslide, could have been caused by an earthquake,
Which we get a lot of here.
which there are plenty of in
Yeah.
the region.
There are a lot of earthquakes.
Mm-hmm.
Yeah, they're right on like two major plate boundary faults.
Like that big one in Venezuela. a few
That
weeks ago.
connects.
It literally
cuts
uplifted
through.
part of the coast 30 feet up in the air. So there's
Yeah.
all these videos from south of Trinidad, where like big rays and stuff just suddenly
Are dead
Are
because
dead.
they were now became part of it.
in the air,
Yeah.
basically.
Felt it.
No, and we didn't even feel it
No, that's
in the
crazy.
country. Yeah, it's crazy.
They think it was the
Oh,
aftershocks.
what's this coming up here?
There's some kind of really gossamer looking coral.
Gossamer.
Oh,
Look at
this.
you. It's a
What does
great
that even
word.
mean?
It's like a fabric, delicate fabric. Old
Excuse me,
timey.
sir.
Look at him.
Yeah.
How do you name
Busting
him?
out with the gossamer.
Quick zoom. Maybe a chrysogorgid? Yeah, but a big one.
Yellow spot hiding.
Yeah, so here we can see a bamboo coral on the left, sponge in the middle, squat lobster in the middle, and then a chrysogorgid on the right.
So something different about here too is it seems like all the squatties are all white, but most other places look red. You know why that is?
So these immunodopsis squat lobsters, and I think they're the more common-ish ones, whereas in my experience, the orange and red ones tend to be living more on or around coral. At least that's my experience. That may not be the case. Whereas I think the white ones tend to be the more ubiquitous, like can live in many different environments.
You said 240 right now.
Yeah, roughly 240 and whatever.
And so even though we've moved away from the seeps, we can expect that the sort of zone of influence of the seeps like probably extends to out here. In the seeps, we're seeing a lot of productivity, a lot of food and life being created, bacterial mats, animals that depend on the bacterial mats or have symbiotic relationships with bacteria, a lot that's growing abundantly and largely. And so that will, of course, then percolate outwards from the seep. So we're seeing some shells of animals that may have been carried out here to be eaten. We can expect that perhaps there's still also influence within the sediment, even though we're not necessarily seeing like any direct seepage here. The influence from the methane seeps will likely still be happening.
Yeah. Yes. Of course. One goes black.
Larissa, can we move this map up a little bit so that we can see sort of down? If we like tilt it a bit. Oh man, more rocks. Yeah, exactly.
Wait,
Perfect.
zoom in on that.
So if we could go left a bit. So that's a big brazingid. These are sea stars, and they filter feed. We can also see, oh wow, oh more nets. We can also see some bubblegum corals again, some sponges, and then this is what happens so often in the deep sea. Despite the fact that we are exploring a place that no one has ever been before, a place that we think is out of sight and out of mind, and also out of our reach, it absolutely is not. And here we can see what looks like a fishing net caught on this carbonate structure and amongst these corals. And we can actually see a dead coral below that was likely knocked down by the fishing net. And it just is a reminder that there is no place on this planet that we as humans have not had an impact, unfortunately. Yeah, lots of sponges.
Well, that says that the red one might be corallium or similar genus. Corallium.
Okay. So we've just heard from we can actually see some corallivery happening. So this is a sea star that is actually eating its way up this bamboo coral. And you can see now how the stalk is actually naked. The branches and stalk are actually naked where the sea star has munched along and eaten all the tissue and polyps away. And again, lots of fossilized coral around. So we did hear from, keep going. We did, once you get a good shot, we did hear that, we did hear that from a car. Wow, look at all these corals behind.
I'm gonna go straight by going sideways.
Here. We did hear from a coral expert, Odalisca Briri from the Universidad de Costa Rica, who has had a look at the images of what we thought was a bubblegum coral, and she has said that actually she thinks it's a corallidae coral, so a different family. Still very cool, still something we haven't seen yet. So these actually look like primnoids. Yeah? Yeah. Yeah. So these, again, these look like primnoids to me. Girl. We can't see any of the bamboo notches moving up on the polyps. I don't know. We're not gonna, let's just not say. This is a deep sea coral. We aren't sure what family it is. It could either be a bamboo coral, so Isididae, or primnoidae. And actually, now that I've said it, I do think it's a bamboo coral again. But we'll see. So yeah, you can just see here how all of these corals are making the most of these substrate patches, these areas of hard substrate to attach to. And the difference that having a little bit of substrate in these sedimented environments makes, it totally transforms what animals are able to be present versus not. So yeah, I guess this is the scarp.
Yeah, this is definitely the scarp. 100%.
Do you want to talk about scarp? You don't have
Sure.
to.
Yeah. Or
Oh
trash.
yeah, some.
Garbage bags?
It looks like a plastic bag. It was just below us. It's okay. Yeah, right there, about to go through the black. But that's okay. We don't need to see it. No. Wow, this is incredible. Yep, that's okay.
We're going to get a little rock sample too.
Are we?
Can we?
Sure.
Of the dark.
You want to take over and direct?
Sure. We can talk about the scarp while we're doing it.
Okay. I think Juliana was meant to be
Oh,
next, but that's
okay.
okay.
Wait,
Are you
wait.
next, Juliana?
Yes, but
No,
Rachel
so
will go
put
first.
Rach, yeah.
Sure. You can talk about it.
Thank you, sirs.
Thank you.
So let me do handovers. So basically, we're not doing 2. We're going directly to 3. We have a little hit list.
But I checked off the ones you gave me earlier.
Yeah, that's great. Thank
Okay.
you. And then a rock. So in the remaining 2 hours on the seafloor, we want to get to waypoint 3, ideally, or until you're pretty satisfied with the scarp. And then
Go up and get soft stuff.
And then, exactly, get some soft sediment, get a heat flow. If we don't make it back to get these to the seep, that's okay. We'll get them in another seep. If we do get time to get a rock and corals, then that. But the things we still have to do are heat flow, cores, and then any other sampling we can.
Okay. Well, while we're in this pile of dark rocks, if we could, I don't know how easy it is to get a sample. I'm assuming you need to settle down. Yeah, which may not be easy, but any of them.
All right, Rich, go ahead. Come into stuff.
That was methane that was trapped under the soil.
Yeah, that's cool. Did you see that, everybody? That was methane under the sub. It was a big burp.
So you want a small rock.
Yeah, nothing, that's going to take too much weight and add it to the porch, but just a sample of these darker, what I assume are carbonates, but want to verify that they're not more of an asphalt kind of.
Okie dokie, I'll tell you what.
I'll let you pick one.
Hop in here and then
Okay.
he can decide where he wants to land out.
Cool.
Right on to sediment.
All right. Thank you, Zach.
We also are very,
Thanks, sir.
Hello.
Where do you want this rock, Rachel?
That is a good question.
You have one call for the left,
I mean,
or the huge Bible.
but doesn't that have a sea cucumber in it?
The Bible?
Biobox 1? I think.
I've got it down as empty here, but we
Uh-oh.
we were this one.
Okay, let me check. Do we have a sea cucumber in Biobox 2? Sorry, 1A? Just making 1A. Okay, there's a cuc in there. We could maybe get a small one and put it in a quiver, or
Last quiver. You want to use it?
okay. Oh, shoot, I don't want to make that decision.
Can it go in on one of the mussels?
Or Biobox 3B that has a plastic bottle in it.
Oh, let's do that.
Yeah, because rocks and bottles get along; they don't contaminate each other.
pores and depot, so trying to get close up as short as possible. Yeah.
All right. So hello, Diva
He's probably gonna want to.
is chatting about important things, and I'm taking over. This is Rachel Lauer. I am a geophysicist from the University of Calgary, and we have had a very exciting dive today. This site is an active seep. We are on the extent of it that is clearly not as active, but if you just notice when we pause the sub, stop moving forward, a bunch of methane bubbles that were trapped under the sub rolled out in front of us. So there's definitely methane in this area as well. I know, sorry. If you see one, it doesn't matter which one it is. It's just one that we can. Yeah, it's hard to. So this whole area is basically if you think about a landslide on land, this is very similar except underwater. So we are at the top of, or an area where that was a massive release of material that slid down slope. And when it did so, it exposed methane hydrate, which has now made its way to the seafloor and allowed all of the chemical reactions and microbial interactions that take place to create all of these surfaces that we're seeing for life to find attachment points and begin colonizing. This is, again, this whole area is a massive accretionary prism. I think it is the largest accretionary prism on the planet. And it extends from to the south of us all the way up through Barbados. And in parts of it, not this part, we're about probably less than 100 kilometers from the toe, but closer to Trinidad and Tobago, just north of there, that region is about 10 kilometers thick. So that's just piles and piles of sediment that have been scraped off the South American plate as it's diving under the Caribbean plate. And that scraped off material just builds and builds and builds until you get this massive accretionary prism. So most of this area is just sediment, except in those places where you get, what about that little one? Like,
I was wondering, can I break off something on this?
yeah. Or it doesn't have to be big. It can be, of course, I don't know.
Something small here.
That little one? Yeah. If that's loose. And we're just trying to get a piece of the rock itself so that we have a sample of something that we can look at in the lab We may get a bonus coral in there. It's probably some huge thing under there. Oh, looks loose. Oh, it's bigger than we thought, of course. Oh boy. Well, okay. Do you think that'll fit in the small one? Might be too big. I mean, we could put it on the porch. It would probably stay there, but.
That should fit in that bottle.
You think? Okay, cool. The BB3B just has a plastic bottle in it,
Yep.
so we won't be hurting anything. So yeah, these are not volcanic rocks. They look just like basalt in terms of color and texture, but the basalt that is what makes up most of the ocean crust is really, really, really far below us. So probably like 4 or 5 kilometers below us. So I suspect this is a carbonate rock that is, I mean, this whole time we've been here in Trinidad and Tobago waters and even further offshore, the only rocks that we've seen are carbonates, and that's because the rocks that make up the crust and all of the volcanics that you normally see at the seafloor, those are all buried by this massive accretionary prism. So the only rocks that form here are either from seeps of active seeps of hydrocarbons, which form these really weird looking, people are calling them asphalt, or rocks that are chemically and biologically or autogenically formed in place through these reactions of chemistry and microbes that ultimately form carbonate. Oh, there's more trash. More human stuff. Yep.
Rachel, do I look? I mean, do I use rock as a regular sample?
It's just in I think it's under geology, but it'll be a normal sample
So
number.
it's number 3 or 4?
I think so. We haven't done one since I've been on the hot seat, but let's call it, yeah. Ask. So it's 304? Okay. Yep. Cool. So we are now on what is called a fault scarp. So if you have just a smooth slope that is, gently getting deeper, at some point something caused this slope to fail and expose a giant methane seep that led to the development of all of these structures that we're looking at and flying over right now. I love the defense of rocks. Yes, rocks are important. Also, some of these are, I don't think we're doing it. Maybe I'll bring that sample back to the University of Calgary and look at, not me personally, because I don't do this work, but there's lots of labs there that can use isotopes to determine exactly when that rock formed. And that will tell us that rock, the formation date of that rock, sometime before that is when the slide happened. Okay, so a good zoom on which this. Can we just get a zoom on this beautiful glass spongy thing? Yeah. What kind is it? I know it's a glass sponge, but oh, okay. All right. It's a beautiful glass sponge. Beautiful. Wow. Yeah, it's a glass sponge, and we're not sure. I haven't seen one like this.
So the sponge people wants this.
They want the sponge, the whole sponge.
I think so. You want the whole? No, just half of it.
Okay.
So just like this.
Can we get a piece of this sponge? Maybe the top half. Wow. Okay, this is after 3 days of seeing mud. This is a very exciting day for everyone here. You might hear the excitement. That is a very large coral. Octocoral. Octocoral.
All of them are octocorals so far.
No, we'd be using oxygen isotopes. This is too old for using carbon dating, but I mean, there's a bunch of different methods that we use for the deep ocean. We use them for dating volcanics, specific methods, and then for carbonates, a different method. But yeah, just to sort of you can't pin it down to a day, obviously, but you can pin it down to millions of years, thousands of years. I think given the amount of, the massive amount of carbonate structures that we're looking at, and just the ages of these, I mean, you think about it, how old are these corals? So some of these corals are thousands of years old, and this rock had to be there for them to attach to, so you have some kind of constraints on how old it is. So they are going to sample a piece of the sponge. They do regenerate. So, wow, that is gorgeous. Wow. Cool. Okay. That is so cool. Oh, okay. I'm not a sponge person, but that's really cool. What's going on in between those bulbs? What's the little... Oh, it's just sediment? Okay. That's wild. I am not sure we know what it is. So we will take a sample. It will not destroy the sponge. They are way smarter than we are and better at regenerating. So, I mean, smarter is a strong statement, but they are better at building things. So, and as I've said before on this cruise, we actually study glass sponges to be better architects. Material science people study them and their spicule design. So this one will be fine, but we will be taking a sample to study it.
So, right.
All right, I'm gonna get out of here. No,
okay.
it's okay. I'll let Giuliano in.
Oh, okay.
Yep. Okay, well, it's been lovely talking to you. This is very exciting. I will see you all, or hear you. No, you'll hear me later. Sorry. Sleep is a thing that we're not getting when it's this exciting. All right, we got enough glamour shots, or?
I think so, yeah.
Yeah?
So we'll take a Niskin. And
Okay. Okay, cool. So you want a Niskin before?
Yes.
Okay. Hold on. We have only 6, I think. I think this is the last Niskin. Okay. So that will be 305.
Yeah.
Ready?
Yes. It went. Cool. Handing it off to Giuliano.
Good. Hi everyone. Hola a todos de la comunidad hispanohablante. Hi everyone, this is Giuliano. And I'm gonna be with you for the next hour. Por aqui estarei na próxima hora. Também para todo o pessoal do Brasil, boa tarde. Vamos estar aqui na próxima hora. Estos brothers, ya. ¿Quieren grabar esta esponja o no? La vida. Entonces vamos a conectar un pedazo de esa esponja y de ahí le seguimos. Le damos pata. ¿Y dónde vamos a poner esa esponja? No es parte de esta vida. ¿En el 4? Is the sponge going on 4? That's the only one left. The last one. Just a piece of the top. I like.
Come from the next side. You're gonna block yourself. Come from the next side. Look at this camera. So fine. We come closer to you. What you need to do now is rotate your fingers so you can see the back. Right now you see it. Yeah, like that. Then come in. You can see the back. So your reference is the looking good. Yeah. You have to come backwards a little. Down. Much nicer. Is this enough for you?
Yeah, that's good.
That's okay?
That's good, right? Yeah, that's good.
So when you close now, you need to rotate the jaws a little so the cutters can kind of cut it. Okay? Like this. So make sure it's closing so the blades are.
Good job, honey. You could feel the tension in the room. It was a delicate operation. Can we get a close up in the arm before we put it away, please? Yeah, thank you. You got that, Charlotte?
Yeah.
Thanks, sis. Beautiful. Después de que guardemos esta esponja, seguiremos nuestro camino hacia el punto número 2 apenas. Para los que han estado aquí todo el día, hemos tenido un buceo increíble y apenas hemos avanzado. After we collect the sponge, we'll continue our way to our next draw point. And for those who have been here all day, this has been an incredible dive, so we have barely moved forward in space. There you go. Thank you.
What number is that?
You come in
306.
That was an awesome collection. Or just go up. I mean,
All right. Thank you, Manny. So I think we can now continue to the waypoint. Just sail away. Solo recordando que una de las grandes capacidades de las esponjas son de regeneración, entonces ese pedazo que colectamos en la parte superior del organismo va a reconstruirse con el tiempo. Just remembering everyone watching, the sponges have an amazing property of reconstructing themselves, so that a piece of the top that we took now will eventually grow up again. Dígamelo.
Nuestro plan original era seguir esta línea, subir acá, pero por cuestiones de tiempo podemos ir subiendo de una vez para tomar los pushers y todo aquí arriba.
Yeah. All right. So our original plan, as I understand, was to follow the line and then go up. But I think for time, if we can,
To waypoint 3.
it was yeah.
Waypoint 3 here.
Yeah. So I think we were gonna like go up here and then come up.
Which waypoints is that you pointed out?
Is that waypoint 3?
Yeah, the one up on the hill.
That's on the hill is waypoint 3.
Alright, so that's waypoint 3 here. So you wanna
So basically we wanna go up directly, like that, so we can start getting some cores on the top of the.
so you wanna come like this.
Yeah,
Or
we can like.
Just straight there.
Yeah, so we can just go straight. No, Beatriz? Because that will give us like this.
It's okay. We just need to be at the top so we can have the sediment.
Right,
Sediment
so
that,
the cores.
plain terrain.
Right, so we want to take some sediment samples, but for that we need to be on the top.
So I have to come starboard, right?
Mm-hmm.
Correct?
Bridge orbit.
Yes.
You want us to go more north?
Yeah, go ahead, RV.
Yeah, we want us to go on
Alright,
top,
uh,
like
you're
kind of like on top of that.
Continue.
Yeah.
Exactly. It doesn't have to be the waypoint.
Doesn't have to be.
Okay, so they wanna
Hey,
come
just
north.
that way, boy.
Next up.
They wanna come north. Not to the waypoint.
Right, because you're not. Where are you in this map?
Look at this map.
This is where we are. You want to come up the hill to waypoint 3, right?
Yeah,
Yeah?
yeah, up the hill to waypoint 3.
Yeah, so that's the most direct route.
That's the scarp.
Yeah, it doesn't have to be waypoint green. We just want to be
So
here.
they just wanna
Oh, so
come
you
north.
just want to come north?
Yeah, we
Up
just want to
here?
come up
Okay.
the hill. Yeah. Thanks.
So I want to get on this scarp.
Delete that station.
It won't delete. Just highlight delete.
No, just right click. Delete.
Okay with that?
So if you just bring in, yeah,
Yeah, just start
more north. Yeah.
Bridge already.
RV bridge.
Hi Tom, so we're going to start heading northwest now, so there's an updated waypoint.
Update waypoint, thank you.
Thanks.
Right, so for those at home, we have slightly changed our route. We need to take some core samples from the sediment, and so we want to move northwest to the top of the fault so we can take the sediment from up there. Because now time is of essence, and we don't have a lot of it now. Yep. Lots of shrimps. Beautiful bamboo coral just passing by below us. Remembering we are 2,000 meters below sea level. Going up a little bit now. And lots of big corals.
Yeah, I understand, but I thought they wanted to get at least somewhere near where
Wow.
they just want to get up to the top.
Yeah, we just want to get up to the top. So it's up. Yeah. No more time.
Those features look so interesting just to the east of Waypoint.
Yeah. They do.
What time you guys have to come in?
We extended it for 3 hours from the original time, so I think we want to come up at 3 now. Yep. Yeah, at 3. I think they want to do some shooting as well, so. Wow. Lots of sponges in here too, and corals. Kind of
Some Trinidad, chicken for bites. Some sea
Whoa. Look at the size of this one. Massive sponge. Little stars on the left. Yeah, massive sponge. 2 of them, actually, we saw. We see some crabs with the anemone on top.
RV Bridge. Hey, are you still heading towards the northwest, or have you moved that waypoint to the north?
Just moved it slightly to the north and then we'll come around to the west of the We're going to stop at the top and take some samples.
Okay, so head for the updated waypoint then.
Yeah.
Some fish, some octocoral. An anemone. No, that's actually a coral as well. More sponges. Lots of them. Big ones too. Oh, look, another one of the sponge that we just collected. Well, at least it seems like it. There's multiple of them, actually. Interesante. Dos de las esponjas que acabamos de colectar. Al menos así parecen. Oh, big fish on the left of your screen. And we're back to sargassum. We've been here for 15 days or so, and I have seen sargassum in every single one of our dives. In a couple of them we saw big patches, big pools of sargassum. To the point that we, a couple of days ago, we collected some samples to understand the carbon sequestration from sargassum, which is quite important for climate change. And so we compared areas of the seafloor with and without sargassum.
Yeah, because we want to reach like the top here.
Like we have like a little one more bump up. We're going northeast. We left the fault and now we're heading back to more of the flat area. Is it now already? You want to know?
Yeah, just want to get everything set up.
Can we, when you get a chance, power the heat flow so he can get everything ready? Thanks.
Heat flow's powered on.
Power on, thanks. Heat flow's power is on. So if we turn a bit west here, will we just go up like that? Is that map true? Because then we can just like jump.
Want to turn west slightly?
Like if we turn left, can we just like whoop?
Okay.
Yeah.
So
Northwest or northeast? Northwest, yeah.
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 So we'll get you to come across here.
Yeah, I think that would be.
290. I'll bring it down. Bridge already.
Harvey Bridge?
Actually, on another waypoint update. Apologies for the changes. We're just navigating our way to the top.
No problem. Update waypoint, cheers.
Thank you, guys. Solo sponge. Here almost looks like part of the sediment collapsed. Well, shrimping away. Corals, sponges. Oh, big coral. Bamboo coral seems to me, from a distance at least, but it's not clear. Octocorals in the middle, and on the right as well. Ooh, so many now. Lots of sponges as well. We can see all the white ones and more corals. Different species of corals as well. A huge one on the left, a sponge. Yeah, that's a big one. Remember that the points, the space between the two laser beams that you see, the two green dots, in about the middle of your screen is 10 centimeters. So as long as they are pointing in the same object, they're measuring 10 centimeters distance between them. Huh? Can we zoom in? Can we? Sorry, guys. Can we zoom in this in where exactly? Just we can start with this part maybe.
Bridge. Or they also.
Coming to a stop.
On the left.
Cylinders, yeah.
Or- Okay. Cool. Yeah, I know. So can you now zoom out, please? Right. And so, ¿cuáles, Beatriz?
They're beneath us.
They're beneath us. Oh, that one? All of this area. Oh, you want this cylindric rocks? Yeah. Well, just like maybe here.
Yeah, it's the rocks, not the animals.
Oh, gotcha.
Do you want to try and pick one up? Just to see what it is.
Rocks, people. We're looking at rocks. Took me a bit to get the memo.
Okay, thank you.
That's it? All right. Thanks, guys.
Bridge on you're clear to continue.
Yeah, we're almost there. Underwater beach. Are you ready? No, but I can.
Overlay. We want the overlay over there.
Oh, sure.
They want the overlay screen.
Just the one right in front of you, Alex?
Yes, please.
And whenever you guys are ready.
This is a good spot.
This is a good spot?
Yeah, we can test it here. Yep, this looks great.
Alright, so guys, can we test here the probe, please?
RV Bridge, I'm just speeding up.
Are we doing a heat probe here?
Yes.
Okay, we're stopping here, Tom, but you can continue until you're safe distance. I'll let you know when to pause it.
Will do, thanks, Manny.
Thanks, Tom. You want the scalper?
Yes,
Yeah,
please.
yes, please. You want to take over? No? We're now gonna do a heat probe test, but before that, we're gonna take out this caliber to see how soft the sediment is here. So we need the sediment to be soft enough so that our heat flow probe can penetrate it. It's quite hard, but still we want to make sure that it doesn't bend or damage the equipment before we deploy it. Vamos aquí a utilizar un equipamiento que mide la temperatura en el sedimento, pero antes de eso tenemos que asegurarnos de que el sedimento en realidad sea suave para que no se dañe el equipamiento.
What kind of grab is that, man?
I'm just put it in.
That's good, man. What type these you guys have to?
What's their
want?
resistance?
Soft.
Soft?
You good?
Yeah. Anytime you're ready.
Anytime you're ready to deploy the probe. making one?
Just one.
Is there one? 8 minutes.
Correct. Yeah.
Is this improvement? Not the index. Heat? Heat, yeah.
Okay, it's going down.
6. 1, 2, 3, 4, 5, 6. That's where we are.
Oh, there it goes. We're gonna do one of those.
One more after this?
No, we're just gonna do this one.
Just stay here.
Just stay here
Just
for.
stay here for
14 minutes. 7 minutes.
So it stays there and then yeah.
So if you want to cut off this and grab this, you want the T, right inside the T that we have. That's your perfect grab. Then we want to let it go now while we're doing this. That's not holding this up. Promise you that's not holding this up. How about now? I don't want to bend that equal.
Just keep the arm still, just because while we're making the measurement, it actually wobbles the, any motion.
Okay.
Sorry.
Sorry, Manny.
Sorry, Manny. Any wobbling makes the probe, the hole around it bigger.
I think the whole ROV is vibrating.
Oh no, but the arm moving makes it.
Yeah.
If you move the arm, we can see it in the data.
Vibration's unavoidable though.
Yeah, part of it, vibration we can't avoid.
It's warm in here today.
There's like 8,000 people in here.
All right. I will leave you all with Jamie and the rest of the heat probe, and I'll see you about tomorrow.
I'll be great. Just coming to a stop here. That's okay.
Yeah, thanks. Oh.
onto the side of the sub on a track, and that probe is then connected to the sub's junction box, which is then through the tether plumbed into the control room and then our laptop. So we are actually able to see measurements as we're collecting them. And so, yeah, the probe is, I think, the only instrument that is plugged in. Yeah, it is the only instrument that is actually, quote, plugged in to the sub, and then through the sub is connected to the control room. So it's pretty cool. We can see on our laptop, as the probe is inserted into the sediment, we can see the frictional heating along all of the thermistors along the lance. And then that's a good check for us that everything's working properly. And we are collecting those data in real time and then saving them directly onto our laptop. So nothing gets stored. It's pretty cool. We can just communicate directly with our instrument.
get jealous.
We're good?
Yeah, so we can pull the probe out, and then we can do whatever it is. So-
We're not taking another one?
No, we don't need another.
Oh,
probe,
okay, okay.
so just
Perfect.
sediment goes on.
Yeah. We're done.
Wow.
Yeah. Now we can, we're all done. So we can take the probe out, and now we gotta take push pulls. Woo.
So just switch off the heat probe.
Yes. There they go. Fun parts over. And I'm gonna push forward right there. Hold on. Yeah, it's saying 12, but I'm only seeing 10 eggs.
And we'll do push pulls here. Yeah.
Oh, these? Okay. But then why are these numbers? Do we take them?
Because are the ones that are there, but this will change.
Okay.
I mean, it's because they are there.
They're just there. Oh, they're in
but
there, but they
yeah.
weren't like taken yet.
That was a mistake.
Okay.
So,
Yeah. We're doing 12.
12.
12.
I think it's only 12 here.
Yeah.
yeah,
we have, yeah.
all of them.
Yeah.
Oh. Yeah, it's just like here we have unexplored.
Okay.
See?
Yeah. 8. Okay. I see.
You wanna reset it? Yeah.
Any specific point or just?
No, just all right there.
Is it good? Come on. So I'm taking it off. I'm putting it back on.
Yeah, leave it like that.
You want me to take
We
it out
usually want a
next?
little
No,
space.
no, I'm just gonna reset it.
Okay.
Yeah, that's good.
So we're gonna place them all.
Yeah.
Might just leave it for a second because we don't want.
And then yeah, when we do it all, we do want like a little space like that, just like what you did.
Yeah, consider this getting a little wacky around there when we were on bottom.
Yeah, it's true.
I'm gonna come back. Perfect, yeah, and I'll
There's my favorite
look at
guy.
the HDMI after that.
Out it out.
Okay. Yeah, if you want to.
Yeah. It's a good day. That's good. Yeah. That's probably like the right amount of space. Yeah.
You want? To the right to see from camera. The camera on the right. Alright, good. Quickly, I haven't seen it there. Alright. Of course, doesn't look straight though. No? It's good. Yeah. Take another? No, no, no. Yes, you're right. Look at that. Sorry. Let's try to get the others straight. Alright, let's keep back up. Alex? What's up? Alright, let's keep back up.
It's looking all right.
That looks practical.
Yeah, you can try it again.
What's that?
than before.
Eat down. 4 more to go. Yeah. They're by the quivers.
Right? Yeah. Block this up. What? Okay. Oh, so if you bring it down in front of the camera, it should be this way.
This?
Yeah. See that people camera instead of that. Really? Two? You wanna see how? Take it out now.
All right, so as we're wrapping up these push cores here, I believe we are going to be off bottom after this. I will be keeping the cameras rolling, so we'll see what we get on the way up.
I think it's enough. A bit discard.
But we've still got a couple push cores to put away,
Yeah,
so
they only need 10
sit tight.
centimeters. That is. done.
You hate me.
Apparently.
Harvey Bridge.
Hello, that's tide complete, so that's ROV off the bottom.
RV off the bottom, cheers.
Thanks.
All right, as you just heard, we are going to be headed back up. So, like I said, we'll leave the cameras running. We've got more than 2 kilometers of water to go up through, so we'll see what we might encounter.